Everyone. Welcome back to the Informatics Institute, the PowerTalks series. Today, before we get started, I would like to just do a quick advertisement for our ATIS meeting, which is our annual informatics meeting coming up on April 22nd, Friday. And for that meeting, if you want to submit an abstract, we have until the end of this month to submit it. I'm going to put the link for the meeting abstract submission and program information in the chat box. Hopefully you can spread the words. The meeting will feature 2 keynote talks. These are very high-profile genome informaticians, 3 sessions and 2 tutorials on AI and the use of Docker containers. It's entirely free, but you need to register in order to secure your spot for some food, catering, and social in person right after the meeting on April 22nd. Okay, well, and then I'll switch back to introduce the speaker today. We're very happy to have Dr. Qianqian Song, who is an assistant professor from Wake Forest University in the Baptist Comprehensive Cancer Center, to give us a lecture on a very interesting and hot topic on informatics research for precision oncology. When I first got to know Dr. Song, it was in AMIA, San Diego last year. And she has a lot of energy, and she networked with many of the leading experts. And we started just talking, and then I requested a copy of her CV, and I was completely taken back by her high accomplishment. She has more than 30 papers published, and this is only her first year as a tenure track faculty. And she has a Ph.D. directly in computational biology, bioinformatics, but actually her background in bachelor and master degrees are in applied mathematics. So I was thinking I was getting a very good education in bioinformatics by doing something like a combination of biology and computer science, but she actually did it the harder way. And how often do you see a young, energetic women scientist who does mathematics and then bioinformatics and cancer research? That is truly a unique combination, and hopefully all of us are just excited to hear the interesting things that you're doing. Welcome, Dr. Song, and it's all yours.
Okay, thank you very much. Thank you very much, Dr. Chen, for the wonderful introduction, and thanks for inviting me to present in this seminar. And today I would like to introduce my recent work of data-driven informatics research for precision oncology. So my work includes the cell-level interrogation of disease mechanisms using the single-cell multi-omics data and the spatial transcriptomics data. I have been working on developing graph-based deep learning models for the alignment of cells across single-cell omics data from the different sources, and the deconvolution of spatial transcriptomics data for higher resolution. During the COVID pandemic, I'm also working on categorizing cancer patients within the National COVID Cohort Collaborative to investigate the risk factors of COVID-19 breakthrough infections and their severe outcomes to achieve the goal of precision care of cancer patients during the COVID-19. So first, I would like to introduce one of the work on single-cell omics data using Graph AI, which has been published in the Nature Communications. In this work, we proposed the single-cell graph convolutional network model to transfer the cell-level knowledge and the cell annotations, cell labels across the single cell omics data. So we all know that there is a fast growing single cell omics data resources currently in the public repositories, leveraging the current known data sets and transfer the cell labels and knowledge to new generated data sets will empower the exploration and the integration of single cell omics data. While the current label transfer methods have the very limited performance, which is largely due to the intrinsic heterogeneity among the cell populations and the extrinsic differences, because they are from different data sets and maybe from different labs, different protocols, etc. So there is a definitely need for an effective label transfer method. So in this work, we present a robust graph artificial intelligence model, single cell graph convolutional network. We name it as SCGCN to achieve effective knowledge transfer across different data sets. Here we name the dataset with known labels as the reference and the new dataset without labels as the query data. So SCGCN is able to accurately identify the cell labels in the query data. So first, SCGCN will learn a hybrid and sparse graph of both inter-dataset and the intra-dataset of the cell mappings using mutual nearest neighbors of the canonical correlation vectors. The dashed line represents the similar cell mappings between the reference and the query data, while the solid line represents the linked cells within the query data. Based on this constructed graph, we use the semi-supervised graph convolutional network to project the cells from both reference and the query data onto the same latent space, so that you can see that the cells with same labels present in the same population. In this way, the cells from the query data can be projected and identified based on the reference data. Here, I want to highlight our graph construction strategy that builds a robust graph based on the canonical correlation analysis, which can identify the shared sources of variation across different data sets. Here we use the canonical correlation analysis across the reference and query to identify their shared information. We also use the mutual nearest neighbor that can identify the biologically matched cells between the reference and query data. Moreover, our construct graph is a symmetric hybrid graph with edge not only link the reference and query, but also we link the cells within the query itself, which can emphasize the similar cells within the query data and the topological relations in the query data. So SCGCN, to show the performance, we have shown the good performance of SCGCN on totally 30 single cell omics data sets. It has consistently demonstrated high accuracy in different scenarios. We have tested the performance on data sets that are from different sequencing platforms, including the 10x platform, SmartSeq2, CellSeq2, etc. We have tested almost all available techniques. We also test the data sets from different species. For example, we may have the reference data profiled from the human tissue and the query data profiled from the mouse tissue. So, or vice versa. Moreover, we show the SCGCN with superior performance when transferring labels across different omics. We have the reference data as the single cell RNA-seq data, and we can identify the labels of the query data from the single cell ATAC-seq data. We also compare the SCGCN with the other methods, including the thread-v3, which is the well-established, widely used toolkit. And we also compare with CONUS, the graph-based label transfer method, and SCMAP, based on the maximum similarity, and the CHETA, which is based on the classification tree to transfer labels. In this study, we also compare with other graph construction approach, such as the cell harmony and the GA models, such as graph search and graph attention models. So we have done a very comprehensive comparison with other available methods. Next, I would like to focus on introducing the comparisons of SCGCN with the other 4 methods, including thread-3, CONUS, SCMap, and CHETAH. First, we compare the performance of SCGCN with other methods based on 12 cross-platform data sets. From the figures, each point represents the accuracy score of the query data. The dashed line represents the equivalent accuracy between SCGCN and the compared method. The dots above the dashed line represent that SCGCN outperforms the corresponding method on these data sets. Based on all these 12 data sets, the accuracy score of SCGCN has a mean accuracy of 87%, which is much higher than the other methods. Of note, when annotating the PBMC data using the cell-seq data as the reference, and we try to annotate the query data of the 10x profiled from 10x version 3, SCGCN is the most distinctively superior than the other methods. The left figure shows the cell labels of the reference data, and the right figure shows the labels identified in the query data. Although these 2 data sets are generated using the different platforms, the figures show the very highly consistent transfer labels by SCGCN for the cell populations. Moreover, from these 2 figures, you will observe that the reference data from the cell-seq is much smaller than the query data from the 10x, suggesting that the effectiveness of SCGCN, even when the reference data is very small, we can also predict very accurately for the query data. For this case of using cell-seq as reference and learn the labels of 10x data, we further show the heat map of the accuracy matrix of each cell type, which are identified by different methods. So the heat map on the upper panel shows the accuracy matrix of each cell type provided by SCGCN, and the others shows the heat map of the accuracy matrix provided by the other methods. And we can see that the SCGCN achieves the best accuracy on each of the cell type. Here, the y-axis represents the true labels and the x-axis represents the predicted labels. And you can see for each cell type, they have very high accuracy for the B cell and the monocytes and different T cell subtypes. After the evaluation across the different platforms, we also evaluate the performance of SCGCN across different species. Here we present the UMAP projection of 4 paired cross-species data sets based on the aggregated data by different methods. The first row represents the human data as a reference, and then we annotate the cell labels in the mouse tissue. This consists of totally 11 cell types. The second row shows the mouse data as a reference, and we learn the labels of the human. So the UMAP projections suggest that the cell labels are aligned better by SCGCN than the other methods, and also the raw data as well. We further present the UMAP projection of the other 2 paired cross-species data sets, which are profiled from the cortex tissue and consists of even more, including totally 33 cell types. The first row shows the human data as a reference, and then we use this as reference to learn the labels of mouse data, while the second row shows the mouse data as the reference to learn the labels of human data. The UMAP projection suggests the aggregated cells by SCGCN aligns better than the other methods. To quantitatively measure the performance in these 4 paired cross-species data sets, we calculated the accuracy score for each method. From the bar plot, we can see that SCGC has the highest accuracy over the 4 cases, and almost around 80%, while the Connors and V3 they all show lower accuracy than SCGCN when trying to transfer labels from different species. Here we continue to measure the effectivity of SCGCN in the scenario of cross-omics. Here we use 4 data sets with paired single-cell RNA-seq data and single-cell ATAC-seq data, which are profiled from the lung cancer cell lines, the brain tissue, the kidney, and the lung tissues. We use 2 evaluation metrics. One is the batch effects entropy, shown in the left panel, and the other is the silhouette index, shown in the right panel, to evaluate the level of cell mixing across the data sets and the preservation of local structure within the data set. The SCGCN presents better mixing entropy than other methods on these 4 data sets. And it also presents a higher silhouette coefficient than the other methods on these 4 data sets, which suggests the better performance of SCGCN when aligning the single-cell RNA-seq and single-cell ATAC-seq data. Here, I want to briefly mention that the batch mixing entropy shows the mixing level of cells in the aggregated profiles of both reference and query data. A higher entropy means a better intermingle and the mixing of cells from different batches, where the single-cell RNA-seq are regarded as one batch and the single-cell ATAC-seq data is regarded as another batch. The silhouette coefficient quantifies the separation of different cell types by calculating the silhouette width of each cell type. So a higher silhouette coefficient represents more differences between cell types, while fewer differences within each cell type, which means that the labels identified by SCGCN is better. To further visualize the joint alignment of single-cell RNA-seq data and the single-cell ATAC-seq data, here we show the UMAP projection of the aggregated lung cancer cells with labels. The cell populations with 0 hour, 1 hour, and 3 hour after drug treatment are clearly discerned in the UMAP projection. And in contrast, the 3 are not able to distinguish these different cell subpopulations very clearly. Based on this aggregated single-cell RNA-seq and single-cell ATAC-seq data, we further identified the overrepresented DNA motifs in the 3-hour specific accessibility peaks, with FOL3, REB1, and ELF1 motif as the most highly enriched motif. This motif also exhibits upregulated expressions in the single-cell RNA-seq data cells at 3 hours. You can see that the FOL3 have higher expression at 3 hours compared with 0 hours after the drug treatment. Similar for the REB1 and the ELF1, suggesting their potential regulatory functionalities after the drug treatment. So after showing the above results, which demonstrate the good performance of SCGCN, here you may wonder whether this good performance is dependent on the selection of the dataset. To verify that SCGCN is robust to the reference data or the query data, we use a well-recognized benchmarking collection of single-cell datasets, which covers 13 major platforms. You can see that the CellSeq2, which is usually used, commonly used as SmartSeq2 and 10x and DropSeq. And we completely examine all possible reference query combinations and that will allow unbiased evaluation of the model performance. As shown in the figure, we randomly pick one dataset from the 13 platforms as a reference, and the others as acquired data. In this way, we get 156 different combinations of reference and query data from all these platforms. The accuracy score of SCGCN versus thread V3 on these 156 pairs are shown accordingly. Across all this data, SCGCN shows an average accuracy of 76%, while thread V3 achieves the average accuracy at 71%. Specifically, SCGCN shows better accuracy on 121 datasets with an average 80% increase of accuracy. So altogether, these comprehensive results demonstrate the robust accuracy of SCGCN. And we anticipate that such superior performance of SCGCN is attributed to the because it simultaneously utilizes the gene expression features, the graph structures, and the reference labels to overcome the batch effects across different data sets. Next, I would like to briefly introduce another work of using graph AI to deconvolute the spatial transcriptomics profiles. This work has been published last year in briefings in BioInfo. A recent development of the spatial transcriptomics associate the spatial information with gene expression within each spot that provides great potentials to understand the tissue functions. However, for such spatial data, each spot is usually larger than an individual cell. The gene expressions measured at each spot are from a mixture of cells with heterogeneous cell types. Therefore, the spatial transcriptomics data at each spot needs to be disentangled so as to reveal the cell compositions at that spatial spot. In this study, we propose a novel method named as DSTG to accurately deconvolute the observed gene expressions at each spot and recover its cell structures, thus achieve the high-level segmentation and reveal the spatial architecture of the cellular heterogeneity within tissues. The DSTG approach leverages single-cell RNA-seq data to unveil the cell mixtures in the spatial data. Our hypothesis is that the captured gene expression on a spot is contributed by a mixture of cells located on that spot. Our strategy is to use the single-cell RNA-seq data derived synthetic spatial, which we call as pseudo-spatial. Here you can see we construct the pseudo-spatial from the single-cell RNA-seq data, and this pseudo-spatial also consists of a mixture of heterogeneous cell types. To predict, we use this pseudo-spatial to predict cell compositions in real spatial data through the graph-based models. First, DSTG constructs the synthetic pseudo-spatial data from the single-cell data as the learning basis. Then DSTG learns a link graph of spot mapping across the pseudo-spatial and real spatial data using the shared nearest neighbors. The link graph captures the intrinsic topological similarity between spots and incorporates the real spatial and pseudo-spatial into the latent space of both using this and compose the link graph. Based on this link graph, we also use the graph convolutional network to learn a latent representation of both local structures and the gene expression patterns, which can explain the different cell compositions at each spot. So this method not only demonstrates superior performance on synthetic spatial data, but also effectively identifies spatial compositions of cells in mouse cortex layer, in the hippocampus slice, and the pancreatic tumor tissues. First, we show the spot deconvolution of mouse cortex layer by DSTG. The left figure presents the UMAP projections of single-cell RNA-seq data from mouse cortex tissue. Different cell types are labeled and colored according to the known cell annotations. This single-cell data is used to deconvolute to the spatial spots. Here, the red figure shows the spatial spot plot using the pie chart, which you can see the predicted cell compositions of each spot within the cortex layer. In this upper panel, we show the proportions of different neuron subtypes, including the cortical layers, L2 and L6, as well as the oligocells. The red spots indicate a higher proportion of the respective cell type, which means if you see the spot has the red color, which means this spot is majorly the oligocells. If this spot maybe have lighter, maybe not that red, it means that the composition of oligocells in this spot is not very pure. The expressions of the cell type markers are consistent with the layered architecture within the cortex tissue, which indicates the accuracy and the sensitivity of DSTG to identify the spatial cellular compositions of each spot. To further demonstrate and test the performance of DSTG in such conditions, we applied it to the spatial transcriptomics data obtained from the tumor sections of the pancreatic ductal adenocarcinoma. The sample matched single-cell RNA-seq data is used to generate the stool spatial as well, which shares the similar characteristics with the real spatial data. For this sample, after the DSTG's deconvolution, we observed a very discrete regional enrichment of cancer clones and non-cancer cells, which is shown in the left figure. And this figure on the right panel shows the predicted proportions of these different cell types, including the antigen-presenting ductal cells and the different cancer clones. You can see they all have different proportions within different locations. Not only the spatial data profiled by the 10x Viron, we also demonstrate the utility of DSTG in the recently available SLASIC platform. They have provided the spatial data of the mouse hippocampus tissues. The typical median library size per bead of the SLASIC platform is 100-fold lower than the 10x Genomics Viron. Here, we also show that DSTG demonstrates very good performance here. The DSTG uses the single-cell RNA-seq data from the mouse hippocampus and to accurately identify the cells within the hippocampus slice of the different cell types. The spatially localized pie charts represent the identified different cell proportions in this slice. Moreover, this figure shows our predicted compositions that provide very detailed information about the heterogeneity of this area. And we also show the closer investigation that confirms the regional enrichment of specific cell types. For example, the oligocells are identified with high compositions in the middle wide strips of the slice, which is consistent with the spatial structures within the mouse hippocampus. In summary, our proposed DSTG method determines the proportions of different cell types and the cell states across spots where genes are reliably identified. DSTG is evaluated on benchmarking data and demonstrates excellent accuracy between the predicted cell mixtures and the actual cell composition. the DSTG is also shown to achieve high consistency with H&E staining observations on the spatial transcriptomics data from complex tissues, including the mouse cortex, the pancreatic tumor slice, and the hippocampus slice, as we've shown before. So the above slides are about using graph-based deep learning models in single cell and spatial data. And I would like to know if there are any questions before I move to the next section.
Qianqian, this is Chen. Excellent work. So I have a quick question regarding the first one, the reference-based. So it's reference-based. I wonder, the reference really determines, right, the classification or clustering results a lot, right? So the label is a clustering label, right? The cell type label, right?
Yes, yes, right.
Yeah, so the question is, if there's a quite heterogeneous tissues or like malignant versus healthy ones, they could be quite different. So is this robust enough to characterize all forms of cell types? For example, in the Thuride or some other methods, they have like different resolutions. They can like switch or they can tune to generate different type of cluster or different number of clusters. Could this method, ICG and GCN, be robust enough to consider all of these issues?
Yes, I think that's a good question. Thanks for asking this. And I think overall, based on the same reference data, ICGC demonstrates a good performance. I think your question may more contribute to the selection of the reference data. If you select a very good or comprehensive reference data with very like customized cell types, I think that it will be no problem of using it to identify the cell types on new data sets based on this reference. So sometimes when you use the transfer label methods, one big question is what reference that you will use. If you use a comprehensive data set, I think there will be no problem. Even there are many internal differences between different tissues, I don't think there will be any problem because this method is designed to overcome that extrinsic differences or intrinsic differences.
And even cross species is okay, right?
Yes, we have comprehensively demonstrated. Let me show you more detail. Actually, I put this as 2 slides because this cross species is from the same data set. You can see that it is from the GSE 84133, and this is profiled from by the same lab actually. We identify the labels based on their profiled human tissue to identify the labels in the mouse tissue. And I also show another study that we actually use the different data sets profile from different labs. So that means we use the human data profiled by one lab, but they are the same tissue actually. Not the same tissue, I mean the same disease. So we use the human data profiled by one lab, and we identify the labels of the mouse tissue from another lab generated. So we also achieve very high accuracy here compared with all the other methods. And you can also see maybe from the same lab, the cross species, there are not big differences as we use it on across the different species and from different labs.
For this one, what are the ground truths? How can you evaluate that the accuracy? What's the ground truth for this one?
The ground truth is we use their identified labels as the ground truth to evaluate the accuracy score. Yes, because that's may raise another question is currently the single cell there is no golden standard cell labels annotated. Yeah, so people always one lab they generated the data, they will annotate in their way, and they will try to make sure all the cell labels are accurate. So we use the published data with the well-recognized labels as the ground truth to evaluate the accuracy.
Gotcha.
Yeah.
Okay, thank you.
Yeah, thank you for the questions. Okay, so I will move to the next section. Okay, I will move to the next session to discuss our recent study of using real-world evidence for the precision care of cancer patients during the fast-evolving COVID pandemic. In this work, we utilized the real-world evidence data to comprehensively explore the COVID breakthrough risk among the vaccinated cancer patients. As is known, there is an urgent demand for real-world evidence on the efficacy of COVID-19 vaccines in patients with cancer. This vulnerable population is very heterogeneous that affected by the COVID-19 pandemic. There are important but unaddressed clinical questions, such as patients with which cancer type are less protected by the COVID-19 vaccine, which COVID-19 vaccine is more effective for which vulnerable cancer population, and which cancer treatment are associated with increased breakthrough infection risk. Our work provides one of the first and largest national level real-world evidence on the risk for COVID-19 breakthrough infections in patients with cancer. We measure the effectiveness of mRNA vaccines in preventing the breakthrough infection. We interrogate the vaccine efficacy in patients with different cancer treatments. And we outline the outcomes of the breakthrough cases. Actionable knowledge acquired in this work will support the clinical decisions for precise cares of the cancer population. The cancer population is highly heterogeneous in terms of the immune response to vaccines and to the infectious disease, especially the COVID-19. Cancer is a heterogeneous disease that is composed of over 100 cancer types. Many cancer types affect specific populations. For example, among the top cancer types, breast cancer and cervical cancer affect the females, and the prostate cancer affects males. The onset age of the different cancer types vary widely too, which determines that this is a very heterogeneous population. The population with cancer is also highly heterogeneous in terms of different cancer treatment that may impact the immunity system. For example, the chemotherapy and the proteasome inhibitors lead to the immunotoxicity and the immunosuppression. B-cell-directed drugs lead to B-cell depletion that impairs the response to virus infections and vaccines. Therefore, the immunity capacity and the responses to vaccines varies a lot among the different cancer types. The cancer treatment history and its impacts on immunity further affect the patient's response to the COVID-19 vaccines. Meanwhile, the COVID-19 is a fast-evolving disease featured by waves of infections and by the fast-changing compositions of the infection, of the composition of virus. As shown in this left figure, for example, in the United States, the alpha variant quickly dominates the infection in about 3 months. As shown in this right figure, the Delta variant took only like 1 month to dominate. Such dynamics is also very geographic specific. For example, as known is the disease evolution at the South Africa has been different, with the Beta variant dominating last winter's peak. As the Alpha variant dominated United States last winter's peak. So it's very geographic specific. The Omicron variant has dominated the new wave, and the one concerning pattern of the evolution of these different variants is the speed up spreading of the new variants. This raises the new needs of the clinical research to adapt to such fast evolving pandemic status. So in summary, the cancer population is highly heterogeneous during the fast evolving COVID-19 pandemic. Precision care of the patients with cancer is a burning clinical need. So a comprehensive analysis on a large cohort in the real world scenarios is crucial to understand the risk and outcomes of cancer patients. We here in the N3C, we define our breakthrough analytic cohort as patients who have received at least one dose of mRNA vaccine between the December 1st, 2020 to May the 31st, 2021. And we also include patients that have received Moderna or Pfizer. And the cohort includes patients that developed COVID-19 at least 14 days after the first vaccine dose. And these patients have never been diagnosed with COVID-19 before vaccination. In this way, we can define the breakthrough analytic cohort by this threshold, by this criteria. We include the demographic variables, including the age at the time of the first vaccine dose, the gender, race and ethnicity, smoking status, as well as vaccine types, cancer types, and treatment histories. We use the available data to calculate the indicator variables on the comorbidity index, which has been adjusted for cancer diagnosis. This is a conceptual diagram of this study. The whole N3C vaccinated population was screened according to the N3C data released on August 27th, 2021. The exclusion criteria you can see here and here was based on scientific needs. For example, we exclude patients that received Johnson & Johnson because the data complexity. This exclusion also considered the data availability and the data quality. In this way, among the vaccination cohort, we remained 6,860 breakthrough cases, with 2,787 fully vaccinated and 4,073 partially vaccinated cases. Meanwhile, there were about 400,000 in the control group that are without COVID-19 infection records, which include about 351,000 fully vaccinated and 51,000 partially vaccinated cases. We noticed a significantly less proportion of fully vaccinated patients in the breakthrough infection group than the control group. Of note, the 6,860 breakthrough cases include 21% cancer patients and 78% non-cancer patients. We did the multivariable logistic regression analysis with all collected variables. And you can see that the age over 65 years and the current of former smokers and the solid and the hematologic cancers, they were associated with increased risk of breakthrough infections. Males and the non-Hispanic Black were at lower risk of breakthrough infections. For vaccination and Moderna, for vaccination here as that received 2 doses, and the Moderna vaccine also showed lower risk of breakthrough infections. Compared with the Pfizer, we did observe that Moderna have better protected patients from the breakthrough infection. The other independent multivariable analysis results that we include the specific cancer types, including the top major solid cancers and the leukemia, multiple myeloma, and the lymphoma for the hematologic cancers in this figure. Compared with the non-cancer cases, patients with hematologic cancers, including the leukemia, multiple myeloma, and lymphoma were at higher breakthrough risk, which indicates these hematologic cancers also had a greater breakthrough risks even than the solid cancers. Given that hematologic cancers were susceptible to breakthrough infection, we assessed the vaccine protection for each hematologic cancer type. The full vaccination protected patients with multiple myeloma, lymphoma, and leukemia, and lymphoid leukemia better from the COVID-19 breakthrough. And the Moderna showed a stronger protection for patients with multiple myeloma. We didn't observe significant protection of Moderna vaccine for other hematologic cancer types. It may due to the limited sample size, and we will examine it further with more data available. We further evaluated the impact of recent cancer treatment on the breakthrough infection risk of this cohort. Specifically, the cancer therapeutics and the bone marrow transplantation procedures were systematically investigated using the treatment-specific logistic regressions. The use of proteasome inhibitors and the immunomodulators were significantly associated with higher breakthrough infection risk. They have a higher adjusted odds ratio, and the medications with major immunosuppressive effects were also associated with higher breakthrough infection risk. The history of the bone marrow transplantation was associated with less vaccine effectiveness. This cause of the observed association between the cancer treatment and the breakthrough infection risk are very complicated. For example, the proteasome inhibitor, immunomodulator, and the bone marrow transplantation, they are commonly used to treat hematologic malignancies, such as multiple myeloma, which by itself is strongly associated with breakthrough infections. We also worked on the stratified analysis to look more or look for the specific drug categories in certain in each hematologic malignancy where the sample size was allowed. In summary, this study investigates the vaccine effectiveness for the heterogeneous and vulnerable cancer population. We reviewed the hematologic malignancies and the solid tumors demonstrate significantly higher risk for breakthrough infection and severe outcomes. This study also highlights the patients who received the recent immunotherapies or targeted therapies did not have higher risk of overall breakthrough infection risk. Such full characterization of the cancer cohort provides insights into the effects of COVID-19 on cancer outcomes and the ability to continue specific cancer treatments and enables the precise protection of the cancer population. The present work has been published recently in Journal of Clinical Oncology. Priorly, we also examined the effects of COVID-19 on outcomes in cancer patients last year, and now we have ongoing projects interrogating the outcome risk of different COVID variants. This work attributes to our team members with diverse backgrounds, including biomedical informatics, clinicians, biostatistics, and epidemiology that work together efficiently and productively. This brings us to the end of this presentation, and I'm almost run out of time. Thank you very much, and I would like to thank you everyone for your time and attention today. Thank you very much.
Thank you very much, Dr. Song. We have a few minutes for questions. If you have questions, you can raise your hand or you can just directly use the mic. Well, I can start with a question and see the rest may think about some questions, if I may. So you really have worked on a wide variety of topics, and all of the 3 topics that you presented today are very hot topics in each of the field, in single cell, in spatial single cell, and COVID analysis. So we have a lot of trainee here and who are struggling with just one idea, but you're able to come up with 3 ideas and execute it quite well. So how did you come up with this idea? Especially I was thinking about the real world scenario. People may say, well, analyze this data for us. How did you just not only analyze the data, but also come up with new tools?
Thanks, Dr. Chen, for the question. Actually, I'm struggling too, but I think maybe I just because I have spent a lot of time actually majorly on bioinformatics, and I'm very interested in developing tools that can specifically meet the biologists' questions that will that seems very I think I'm just very interested in this area, and I think it can help biologists and move the bioinformatics field forward. And I think it's challenging, but it is also enjoyable too.
Okay, other questions?
Qianqian, I have a quick question regarding the COVID work. I think this is fascinating. Technically, a naive question regarding so there's many factors besides this COVID of the vaccination status or something. So there's the age and some others. So which approach did you use to adjust all of these factors, and what statistics work or approach did you use to adjust or considering all of these factors?
Yes, we use the multivariate logistic regression to adjust to include all potential factors into one model to evaluate the odds ratio of the different factors. Yeah, you can see that you can see the age with different range and gender, race, and ethnicity, and different factors here. And we include all of them into one model and to evaluate the potential maybe compounding effects, to adjust the potential compounding effects to evaluate the real impact of the COVID-19 and the breakthrough infection risk.
So it's kind of a stratification, right, of different tiers of data and compare between groups. It's not a propensity score kind of based approach, right?
Yeah, we didn't use a propensity score in this study because we have considered it, but we finally decided not to use the propensity score because some potential reasons in the N3C platform. And yeah, we will consider also try using it in our ongoing study. But in this study, we didn't use the propensity score yet.
Okay. Another question is, I think it's probably you cannot address. It's just some observation. So the race and the ethical groups, the African American group seems to have a better the breakthrough infection compared to the other groups, right? Why was that? What's the reason behind this? Another is the hematologic cancers, right? These liquid cancers, and they have a worse, even, I don't know, even if the patients have been vaccinated, right? They have even a worse breakthrough infection compared to the other cancer types, right? If I understand correctly. So what's the reason behind this? This is fascinating. I think it's very interesting anyway.
Yeah, thanks, Dr. Chung. You mentioned a very good point because we also noticed that the health disparity, because for people may think the non-Hispanic Black patients have like more risk for the breakthrough infection. We saw that before, but after our results, we found the patients with the non-Hispanic Black patients actually have lower breakthrough infection risk. So we anticipate that it may due to the vaccination rate. Because the vaccination rate of the African Americans was lower than other races. So maybe hesitancy issues, but our observation also validated by some other reports as well on smaller cohort maybe. Yeah, we discussed it, but it is an interesting finding and consistent with the other available report. So we are trying to maybe dig further on this part, but not yet. Yes.
Sorry.
Go ahead, Zhongliang.
Another question is about the control and case, the balance. How do you just balance the number of these control and case and do a fair comparison and generate those significance?
That's a good question, but we didn't adjust the number of differential the differential size of the breakthrough and the control cases. We did the logistic model on all the patients together and to look into the risks of different factors. So we actually not just for the numbers of the cohort.
And also, like as Dr. Chen Chong mentioned, because the controls and case are not balanced, is there any like covariant balance, like if they compare the same confounder effectors to get the real significance using the case and control?
That's a good question. Actually, we are asked by the reviewers too on a similar question, I think, because the reviewers are concerned about the potential confounding effects on this analysis, but we use the different evaluation metrics, including like the different evaluation metrics to demonstrate that there are no confounding effects and every factor, their risk is significant. So our included variables are all make sense for within this model. Not sure if I answered the question very well. Yeah.
Thank you. Yeah, by the way, I'm Zhong Nangyu from the Informatics Institute. Yeah.
Okay.
Thanks.
Thank you.
Well, thank you, everyone. We're at time. Let me conclude by quoting Shakita from the audience. She said, great power talk, Dr. Song, on AI, single cell and COVID-19 as relate to informatics science in hematology/oncology, and then 5 exclamation mark. I think that properly conclude our opinion. Great job, well done. Thank you so much for your time, Dr. Song.
Thank you very much. Thank you.
And we hope that to be in contact and maybe collaborate and collab.
Thank you. Thank you.
All right.
Okay. Thank you, everyone.
Bye-bye.
Bye.